Hello, my name's Tom Greenfield, and I'm going to be giving a basic overview of epigenetics, that is, the way that gene function can be influenced by factors outside the DNA. There are many examples of how this works, but for simplicity, we're going to look at this concept through the lens of one gene and how its function can be altered by external factors. Firstly, we're going to be looking at genetics, which is traditionally taught as reductionistic science. But what do we mean by reductionism? Methodological reductionism is the position that the best scientific strategy is to attempt to reduce explanations to the smallest possible entities. In biology, this means explaining all biological phenomena in terms of their underlying biochemical and molecular processes. If you know the laws governing the behavior of the parts, you should be able to deduce the laws governing the behavior of the whole. Inappropriate use of reductionism limits our understanding of complex systems. An opposing theory to reductionism is that the whole has properties that its parts don't have on their own, or there is something in the whole which cannot be explained by reducing it to its component parts. Science has to find out how to study ways in which larger scales of organization influence smaller ones. This is known as emergence, where the behavior of the parts is determined by the behavior of the whole. One example of emergence is how individual muscle cells work together to produce a structure of a muscle and result in movement. Another example is how individual atoms of hydrogen and oxygen combine to form water, which in combination form the flow found in a river and waves in the ocean. Many different components make a forest, trees, plants, animals, and earth. This is different to thinking about the parts in isolation. We're going to look at some aspects of genetics which are typically considered reductionistic and see if we can fit it into a bigger, more emergent picture. First, I'd like to quickly revise the concept of genetic mutations. When a gene is what we consider normal, it produces a functional protein. Normal usually means it produces the same type of protein that the majority of the population have. When there's a difference in the DNA code to the normal version, depending on the type of change, either a different shaped protein can be made, more proteins than usual are made, or no protein at all is made. If the protein is not made and it's vital to life, then the individual would not survive pregnancy as a fetus, a possible reason for miscarriage and fetal death. If the protein is functional, it might have reduced function due to being folded in a way that's less efficient, or having some parts missing. We can't do anything to change the protein as it's hard-coded into the DNA, but we may be able to support the function of the protein by providing everything that it needs to function, such as specific nutrients or coenzymes, which is how nutritional supplementation can work. We can also support the production of the protein so that even if it has a reduced function, we can get the gene to produce more of them to give this metabolic pathway the best chance of functioning at a level that's needed. So let's look at one specific gene and the protein it makes. Nuclear receptor 3 group C member 1 is a protein produced by the NR3C1 gene, which is designed to trigger the response to cortisol in the body. Activation of the NR3C1 protein by glucocorticoids moves the protein from the protoplasm into the cell nucleus. This triggers the activation of transport or transcription of other genes, which are responsive to cortisol and have an anti-inflammatory effect. At the same time, it also prevents pro-inflammatory transcription factors from being transported into the cell nucleus. The result is an overall reduction of inflammation, which completes a homeostatic feedback loop by signaling the end of a threat response, which reduces cortisol production. The NR3C1 gene is located on the long arm of chromosome 5 at position Q31.3. As well as being involved in inflammatory responses, it also has a role in cellular proliferation or cell division, and also differentiation in specific tissues. About 20% of the genes expressed in human white blood cells are regulated either positively or negatively by glucocorticoids. When the NR3C1 gene contains certain mutations, that is, rare variants that prevent the gene from producing an active protein, this results in a rare genetic condition known as generalized glucocorticoid resistance. Clinical features include hypoglycemia, hypertension, metabolic alkalosis, chronic fatigue, and profound anxiety. Other less significant variants may alter the function of the protein to a lesser extent, so that it might be less efficient. For example, evidence that decreased NR3C1 expression in the hypothalamic pituitary adrenal system is often observed in suicide victims with a history of childhood abuse. Let's revise how proteins are made. The mechanism for reading genes starts with the DNA being unraveled by histones, the string that holds the chromosomes together, relaxing and unwinding them. The DNA starts off coiled up. When a protein is made, histones relax and the DNA becomes available to be read. Then the RNA-making enzyme, the polymerase, reads the gene, starting a specific region, the promoter region, by splitting the double helix into two strands. Transcription takes place by copying the codons into messenger RNA, or mRNA, which acts as a message. mRNA takes this message to ribosomes, which make the protein from the message in a process called translation. Like other proteins, the NR3C1 protein can only be produced by the NR3C1 gene when it's available to be read by RNA. The NR3C1 protein has several isoforms, that is, variations in the way that the gene is read, which make a different shape protein. The different proteins result in variations in the way it's carried from the cytoplasm to the nucleus, and different transcriptional activities. Let's look at mechanisms that can change the way the gene is read to produce more or less of the protein. Now we're looking at what's called epigenetic factors, that is, mechanisms that change the way a gene produces proteins without any changes in the DNA code, affecting both the speed of protein production, silencing or activating it, and the type of protein produced. This happens mostly before RNA transcription. Histones, the string around DNA, can be relaxed to allow the gene to be read by RNA, or tightened to prevent the gene from being read. This is done through several mechanisms, including methylation. There are areas of the gene known as promoter regions which stimulate the production of the gene. DNA methylation in these regions and the first exon of the gene can switch off the promoter function of the gene, which prevents the transcription of the gene. DNA methylation of the main body of the gene after the first exon is associated with high levels of gene expression in dividing cells, although it may have the opposite effect in slowly dividing or non-dividing cells. Let's look at how methylation works. This is the closer look at methylation, how a methyl group, that is the yellow circle labeled CH3, attaches to the cytosine molecule on the right and becomes 5-methylcytosine on the left, and vice versa. Cytosine is an amino acid that's used as a nucleotide that combines with guanine to make part of DNA called CpG islands, a situation where cytosine in the DNA code is followed by guanine, separated by phosphate. Cytosine is the C of the 4 amino acids GACT, and guanine is the G, and along with adenosine and thymine, these form the rungs of the DNA ladder. The cytosine in a CpG group can be easily methylated by a methyl group, a molecule of carbon which has 4 potential poles of attachment with only 3 hydrogen molecules attached. A methyl group is abbreviated as CH3. Here the cytosine amino acids are represented as the green rungs of the ladder. You can see the CH3 methyl molecules, 1 carbon and 3 hydrogen, have attached to the cytosine on the DNA, forming little satellites on the outside. When DNA is methylated, it gets coiled up by histones, complex molecules which pack up the DNA into coils and prevent it from being read. Methylation is one of the triggers that condenses the histones to form heterochromatin, a tightly packed form of DNA. Acetylation with the histone acetyltransferase enzyme relaxes the histones by using the acetyl group, which is a methyl molecule CH3 with a carbonyl group attached, that is a carbon with a double bond to an oxygen molecule. This acetylase enzyme attaches to a lysine molecule at one end of a histone tail, which activates the DNA coiling. As I mentioned earlier, DNA methylation in the promoter region of a gene is associated with repression of transcription, so less protein production, whereas DNA methylation in the body of the gene is generally associated with increased gene expression, that is, more protein production. DNA methylation patterns are passed from one cell to the next during cellular replication. In this picture, the parent strand of DNA is shown in green. and the new daughter strand is shown in blue. The methylation pattern in the parent strand is the grey attachment, and enzymes called DNA methyltransferases replicate the position of the methyl molecule and place it on the new DNA strand. This is dependent on the raw materials for making the methyl donors being present. Therefore, diet can affect methylation through the presence or lack of methyl donors, which include nutrients such as folic acid, vitamin B12, and betaine. Lack of these nutrients reduces the amount of methylation that can take place during cellular replication, which can affect the coat color of the offspring of agouti mice, and can also result in them having diabetes. Crossing a yellow diabetic agouti mouse with a brown non-diabetic agouti mouse resulted in more brown non-diabetic mice as offspring when the mother was fed a diet rich in methyl donors while pregnant, and more yellow diabetic mice when she was fed a diet deficient in methyl donors. In humans, a low methylating diet or a diet low in folic acid can also lead to conditions such as spina bifida and Down syndrome. But there's also increasing evidence that synthetic folic acid supplementation in pregnant women can cause other problems in their offspring. Another thing that can affect methylation are toxins, such as the chemical bisphenol A, which is used to make some plastics. The mothers of both mice were fed bisphenol A. The mother of the small brown mouse on the right was also fed methyl donors. Heavy metals can also affect DNA methylation through epigenetic mechanisms, as well as by binding directly to various methyl response elements in the target gene promoters. So here's what can happen to the baby before birth when the mother is stressed physically, mentally, or nutritionally, or even with toxins. Genes in the embryo, such as NR3C1 and other genes which relate to the HPA axis, are adversely affected by epigenetics, affecting the way glucocorticoids act in the cells of the embryo. This can result in changes in neurobehavioral and cognitive function as an adult. Several studies have found that increased or decreased methylation of specific parts of the promoter region of the NR3C1 gene influence the transcription of NR3C1 in the hippocampus, the center of emotion, memory, and the autonomic nervous system. This alters the cellular response to the HPA axis. This was discovered by a study done on rats when decreased methylation of a part of the promoter region of the NR3C1 gene resulted in increased expression of transcription of hippocampal exon 1 of NR3C1, and subsequently increased negative stress regulation of the HPA axis feedback, requiring less glucocorticoids to be secreted for an appropriate cellular response, and therefore resulting in a more sensitive response to stressors. Here's an example of a study done in humans. This is a small study that was published in 2019, where 30 mothers and children were studied in infancy and early childhood. The children all had developmental problems. The researchers measured the amount of DNA methylation on the NR3C1 gene promoter region of the children at 1 month old. The children which had the most methylation in this area tended to have better social and emotional functioning at 6 months, according to a questionnaire designed to score this. Increased stress reactivity in the hypothalamus pituitary adrenal axis in the children of mothers exhibiting lower levels of maternal care appears to be caused by decreased expression of glucocorticoid receptor gene, that is the NR3C1 gene, in the hippocampus. This is from a 2016 paper where the authors looked at the NR3C1 methylation of placentas of 120 newborn babies, as well as their cry acoustics, various parameters of their cry when they were poked in the foot by an examiner. They found that there was an association between the type of cry that the babies made and the amount of methylation of the NR3C1 gene in the placenta. The babies from those placentas with the most NR3C1 methylation had more of the type of cry associated with poor regulation of stress response. Another paper said, the degree of NR3C1 promoter methylation may serve as a useful marker to determine severity of abuse and determine if those who experience childhood maltreatment are at risk of developing severe psychopathological symptoms later in life. It's not just the NR3C1 glucocorticoid receptor that gets methylated in a stressful situation. Other genes include SLC6A4, or solute carrier family 6 member 4 serotonin transporter, which transports serotonin around the body, BDNF, or brain-derived neurotrophic factor, which is active in the hippocampus and encourages the growth of new neurons, and OXTR, or oxytocin receptor, which influences a variety of behaviors, including stress and anxiety, social memory and recognition, sexual and aggressive behaviors, bonding or affiliation, and maternal behavior. Factors that can affect gene methylation and therefore include not only the availability of nutrients and presence of toxins or heavy metals, but also oxidative stress and psychological stress. In terms of potential physical illnesses, you can see how adverse childhood events and stress can result in asthma, for example, with increasing age. Exposure to adverse childhood events can result in chronic activation of the HPA axis, leading to increased baseline cortisol levels, which affects neurodevelopment and impaired social functioning, creating a vicious cycle of increased exposure to stress and ending with chronic illness. As well as maternal nurturing, other self-care activities can alter the methylation of genes involved in neurobehavior. This study found that a 12-week Iyengar yoga intervention increased the expression of the NR3C1 gene and other glucocorticoid receptors in 16 breast cancer survivors when compared with those on a 12-week health education course. The effect of NR3C1 is anti-inflammatory because it allows glucocorticoids to act on the cells. The yoga course also reduced the expression of many other inflammatory genes. If yoga can do this, just imagine what effect osteopathic treatment might have on gene expression. Let's put this all together as to how osteopathy might be useful in this field. I want to talk about how these metabolic pathways function inside the cell. Each metabolic activity is influenced by the electrical charge in the cell, which enables the activities to take place that are carried out by proteins through reduction and oxidation. Water is present everywhere in the body, and it's known that it can have a structure or it can be unstructured. When it is structured water, it's considered semicrystalline, sol and gel. Crystals can generate an electrical charge, and so water in its crystalline form has an electrical charge. Because chemical reactions use electrical charge in reduction and oxidation, water is an essential part of these processes. Structured water holds an electromagnetic field, which changes its voltage in picoseconds by structuring and destructuring. This allows cellular metabolism to take place more efficiently than if molecules were just floating around without any electromagnetic direction. There's a kind of machinery inside the cell that uses this ability of water to change its voltage. On the right, this is a picture of G-actin, as in actin and myosin. Actin being the protein that forms the basis for movement in myosin during muscle contraction. Myosin moves along filaments provided by actin. Actin is capable of producing filaments in picoseconds and is hypothesized to be responsible for osmotic activity. The Z on the left turned on its side is the F-actin form, a polarizing form, so the particles can come in and out of the cell. And on the right side is the G-actin form, which repels particles such as elements. It allows the shifting of possible states of folded and unfolded protein. This actin binding site is the main binding site. If ATP isn't there, the water can't be structured. Other ways include light or other energy. ATP is a highly polarized electromagnetic element and acts like a magnet to the rest of the water molecule. The G-actin contains magnesium, and this is how magnesium structures water. Calcium is also on the other side. The whole process is an electrical impulse. I don't have any evidence, but it makes sense that osteopathic treatment assists this process by organizing the unity of the flow of fluids in the body. Finally, this is a variation of the classic image by Waddington on the left, who depicted the epigenetic landscape as a series of hills and valleys corresponding to the developmental history of a particular part of the egg. Strings under the valleys are pulled by genes to alter the pathways that part of the egg takes during development as it rolls downhill, pulled by the gravity of time. On the right, we're looking at how manipulation of gene function through elements that influence the epigenetic effects on the egg can push it back uphill to take a different pathway or move it into a different valley. In this way, we're not changing the cellular DNA, but changing its expression by influencing the whole. I hope this has been a useful introduction and overview of epigenetics and its relevance to osteopathy. Thank you.